Review lecture 2 and 3. The title is Introduction to Terminology and Drug Discovery still. And we first talk about what the medicine is. Medicine is a substance used to treat disease. Pharmaceutic, therapeutic, distinction between drugs that are used in medicine and drugs that are abused. So pharmaceutics and therapeutics distinct. The difference between drug and medicine. A compound that interacts with a biological system and produces a biological response could be either drug or medicine. So pharmaceutical chemistry. Is also called medical chemistry or medicinal chemistry. Pharmaceutical chemistry is also known for medicinal chemistry and is a discovery and or design of new therapeutic compounds and their development into medicines. Pharmaceutical chemistry involves in 4 areas. PharmChem involves 1. Synthesis and isolation. 2. Structure, activity, relationships. And we abbreviate it as SAR. 3. Knowledge of interaction. With receptor and enzyme mechanism. And lastly, we abbreviate it as ADME. Tox stands for A, absorption, D, distribution, M, metabolism, and E, excretion, and TOX for toxicity, toxicology. Now let's talk about what good versus bad drugs are. So all medicines have benefits and drawbacks. A good medicine would have do what it's meant to do. Have acceptable toxic. Or unwanted side effects. And be easy to take. And we can talk about morphine here. Morphine in low dose, it is an excellent analgesic painkiller, analgesic, which is also a painkiller in low dose. But has a serious side effect, or it has side effects. And the serious side effects violates the second. It does not have the unwanted side effects. It has addiction and intolerance, and also it causes respiratory depression and may kill if taken in excess. So it has a very serious side effects. So we can consider morphine as a bad drug. And we also have examples of everyday drugs. They are 1. Caffeine. Caffeine is a stimulant in coffee, tea, or cocoa. Drinking coffee is taking a drug. Sugar interacts with the taste buds of the tongue to produce a sense of sweetness. Nicotine in cigarettes, it's a sedation or calming effect. It has a sedation or calming effects. And lastly, alcohol. Difficult to judge the correct dose required to gain the desired effect without drifting into higher dose levels. But that also produces unwanted side effects and impaired judgment. Let's talk about medicinal chemistry, folklore, which is the history of the medicinal chemistry. And all this medicine discovered so far is about 5,100 years ago. In Chinese Emperor Shen Nung, the Book of Herbs, they had a plant called Mahong, and this Mahong was used as a heart stimulant and for nasal and chest congestion. And this nowadays has found to contain ephedrine. Looks like and more therapeutics extract a foxglove plant cited by Welsh physicians in 1250 used to drop C congestive heart failure in 1785. Okay. Now let's also talk about aspirin. This was found 400 BC by Hippocrates. Chew bark of willow tree for pain, for childbirth or eye infections. Active component of willow bark is salicin. Willow tree contain salicin, and salicin and aspirins are considered prodrugs. So aspirin, let's talk about aspirin a little bit. Itself has a anti-inflammatory action, and so it's a pain relief. So salicin becomes salicylic acid, and it becomes acetyl salicylic acid, and that acetyl salicylic acid is called aspirin. It's converted and also converted here. So the definition for prodrug is when you observe something that does not have the effect yet, body converts to a drug that has effect. And structure for salicin, we will talk about it later if it's necessary. Another example, penicillin. Penicillin looks like this. It's a blocking bacterial cell wall in biosynthesis, or block bacterial cell wall in biosynthesis. It has a amide bind in the ring, and it is called lectin or lactone. An ester linkage with the ester linkage. So that was found in 1928 by Alexander Fleming. That he accidentally used this in the bacterial culture that contaminated with mold and saw that it had antibacterial effects. So they were the discovery of some drugs. So where do we get new medicines from? So we isolate them from nature. They are called natural products. So types of natural products are fatty acids, polyketides, isoprenes, alkaloids, nucleosides, peptides, sugars. I'm going to talk about that in a few minutes. Organisms secrete toxic natural products as chemical warfare against each other, like marine organisms or fungi. Marine sponges are good examples that they generate complex molecules to defend themselves. It is like a primary natural products. And we can secondly synthesis. We can get the new drugs from synthesis by chemical and enzymatic. So we make the derivatives of the natural products or completely new scaffolds for the new drugs. So let's talk about some natural products. So the first class we'll be talking about is the fatty acid. It's a form, an example of should have drawn in a little longer. Oops. And where the kink is, there's a double bind. It's a arachidonic acid. It's a precursor to pressed glands or leucorrhins. It's a derivatives influence pathways. So fatty acid is one of them. Another class, polyketides, that is from polyketone. precursor. It has a name of quaternarial from fungus, and it can transform to this, and sulfur that is part of the enzyme is necessary to drug take. Another example for natural product class would be terpenes. And terpene shall look like this, and it has a name of bisabolin. It has a cyclohexane. So basically it's not perfectly plain, and it has a derivative of isoprene, or piezo phosphate. And this product has the name of DMAP. P stands for dimethyl aryl or allyl pyrophosphate. Another natural product class, alkaloid. It contains nitrogen, used as a electron acceptor at specific pKa, lone pair acceptor, and many of these are derived from amino acids. Some amino acid derivatives, and shall look like is a basic nitrogen. That is a Bronsted base and protonated, and also makes soluble in water. And this drug is actually cocaine. And very similar drug that is a derivative of cocaine, we have Novocaine. Novocaine is like cocaine without addiction. Okay, so let's go over some more great features of this nitrogen. It's a Bronsted base. It's protonated and soluble in water. There's a hydrogen bond acceptor. And what's important about soluble in water, it can circulate in blood. Circulate in blood. Other natural product classes could be nucleosides, we probably don't need to talk about it, just know that nucleosides is another natural product class. So about 40% of the drugs approved in the last 20 years are natural products, and the other 60% are the natural product derivatives. Drug discovery process and basic steps. 1. Identify target. So biomolecules as cause of disease like protein or DNA. To identify a compound. And to identify a compound, we do rational design, rational structure based design, and library screening. Lastly, we modify the lead compound. For our compound optimized. So it increases or decreases the potency, and it lowers the toxicity, then generate candidates for clinical trials, balance activity, and ADME tox. Okay, then let's talk about the 1, 2, 3 that I've written here. So as an example of the penicillin, it has a target of the cell wall of bacteria, and to identify a compound, so some molecules that interact with target, which in the very beginning of the process, in rational, it mimic the intermediate, and some previous info. And in library screening, we can see the collections of a lot of molecules. And number 3, when we modify the lead compound optimized, then we can make it better, as in terms of lower dosage and also economic benefit, or lower the chance of the side effects. How do we determine which molecules are useful as potential drugs? Then we will check the correlate structure with biological activity using we check structure-activity relationship and characterize interactions with receptors. And examples for receptors could be enzymes or DNA. And we characterize practical properties. And practical properties again includes ADME-tox, and these are referred as pharmacokinetics. Pharmacokinetics, ADME-tox, very important. So for pharmacokinetics, we don't talk about interactions with the target, not interactions with target. We talk about the features and properties of molecule itself. And later on, we will talk about pharmacodynamics, which is the interaction with the target. So everything that is not the interaction with the target itself, we call it pharmacokinetics. Okay, so this was it for lecture 2 and 3 review, and I will come back for lecture 4 review.